Welcome, my name is Magdalena Iturria. I am Director of Food, Land and Agriculture at Circular Influence. I will share my screen in a moment. So I will be hosting this webinar on sustainable food, agriculture, and land use innovation ecosystems. So I'm sorry, I was told that the video had no sound, but it was just some background music. I hope you got the message from our video. Basically, what we are telling is that the time for action is now. We are running short on time, and that's just the mission of Circular Influence, is to bring together all these different stakeholders from education, businesses, and policymakers, so that together we can build and accelerate solutions and get them quickly to the market, so that we can bring about the much needed transition from a linear to a circular economy. So today we are going to get a glimpse on the wine innovation ecosystem for a circular economy, from land to packaging and back. I will be introducing some key aspects of soil health for sustainable viticulture. My co-panelist, Professor Bruno Mezzetti, will be talking about vine genetics for sustainable viticulture. And my colleague, Arquitecta Alejandra Núñez, will be presenting a very compelling example of sustainable packaging based on wool. So for the first part of our webinar, I will be talking to you about soil health foundations for sustainable viticulture. When we think about soil fertility, what we are usually referring to is to the chemical components of soil fertility, the nitrogen, phosphorus, potassium, minerals, oligoelements that the plant will need. Sometimes we also consider the physical aspects of soil fertility, soil structure, compaction, infiltration rates. But what we usually leave out, and what is probably one of the most important parts of soil fertility, is the biological fertility, the micro and macroorganisms that are actually present in the soil and that are responsible for actually building the chemical and the physical components. There are more microorganisms in a spoonful of healthy soil than there are people on the planet. And nature has done this for a purpose. The soil and the microorganisms in it act as the plant's digestive system. They break down foods and make it available for the plant. They solubilize nutrients. They extract the nutrients from the sand, silts, and clays and make them available for the plants. And it is now known that a healthy soil with the right biology present will be able to solubilize and present to the plant up to 90% of the nutrients that the plant requires without the need for external fertilizer. These microorganisms in the soil also produce different enzymes, hormones, and all different kinds of substances that will be able to provide natural pest and disease control for the plants. And also the interaction between the plants and the microorganisms in the soil will be responsible for carbon sequestration. The plant will go about its photosynthesis, sequestering CO2 from the atmosphere, exudating it through its roots, and then the microorganisms there play a huge role in fixating the carbon into the soil. So recent advances in science and soil ecology have discovered that there is a huge ecosystem below ground. There is actually a whole soil food web down there where millions of different types of microorganisms play different kinds of roles. And the very interaction between all these microorganisms is the one that is responsible for giving all those benefits to the plants. And if one of those links in the soil food web is broken, then of course that the whole benefits of what we mentioned before, the solubilization of nutrients and the pest controls will not be able to be present naturally because the soil food web is broken. And also something that is very curious and very important about this is that the very plants that grow above ground are the ones that are responsible for feeding and determining the types of microorganisms that we will have below ground. It is now known that plants photosynthesize, and about 30 to 80% of what they produce through photosynthesis is then exudated through their roots down into the ground to feed the microbes. So that's just how important microbes are to plants. And also when you have several different types of plants growing above ground, there is a huge diversity above ground. You will be producing different exudates through the roots that will be able to feed different kind of microbes and therefore have a very robust soil food web and able to really perform all the different functions that the soil food web must perform. What happens when we have one single type of plant or a monoculture is that we are just producing and feeding a limited amount and kinds of microorganisms, and therefore the soil food web will be debilitated. Just to make this an example for us, it would be to think about our own bodies and our own digestive systems. We know that to have a healthy gut and to have a healthy body, we need to have all different types of foods and eat from all the different families of foods. And the same thing happens to the soil. If we eat the same sandwich every day, we know that we will be more dependent on vitamins and other supplements and medication to keep our health. And the same thing happens in the soil. This is exactly what's been going on for decades and decades of conventional agriculture, where excessive tilling, excessive input of different chemicals has deteriorated and broken the soil food web, and especially also the monoculture crops have been limiting the foods to the soil, and therefore the soil food web has been broken. Which then leads us to what is obvious. What we are seeing is that we are more and more dependent on bringing external fertilizers, external pesticides, external insecticides to control our crops and to feed our crops, because our soils are not being able to do what they are supposed to do. Our soil food webs are broken because of this. But regenerative agriculture and the principles for regenerative practices are thought to restore this soil food web. They are meant to restore this whole diversity in the soil, and they are able to harness the whole power of all this biology available. So when producers get creative and they learn to adopt these 5 very important principles of regenerative agriculture, they need to adapt this to their very own needs and objectives. And once they learn to do that, they can learn to harness all this power of biology below the soil and be able to produce better and be more resilient with less costs and less need for external inputs. So viticulture in particular has a huge potential to harness this soil biology, to modulate the soil biology by incorporating multi-species cover crops, incorporating a lot of plant diversity along these interrows, where you can modulate the biology, restore the soil food web, and even in some cases also integrate grazing animals to stimulate the multi-species cover crops, to naturally fertilize them with manure. And in this way, the microbial networks that will be developing below ground will be able to provide the vines with the right ecosystem to produce more nutrient-dense foods, to naturally control diseases and pests, and ultimately be more resilient, increase water retention capacity. There are a huge number of benefits that can come out from this. We have a lot of examples of different producers that are now insecticide-free and pesticide-free just by incorporating these 5 regenerative principles and incorporating plant diversity in their ecosystems. So that was just a glimpse of what I wanted to share with you. Thank you for listening. And I would now like to introduce our second speaker for this webinar, the renowned professor, Dr. Bruno Mezzetti, from the Universidad Politécnica de Le Marche. Professor Mezzetti is author of a total of 185 publications and several book chapters, and he has been recently awarded the honor of Officer of the Order of Merit for the Italian Republic. Professor Mezzetti will be telling us a little about his work in genetic resistance for sustainable viticulture.
Yeah, now you hear me, sorry about this. And so I go here back again. So I want to say thanking everybody, thanking Maddalena and the organizer of this concert, giving me the opportunity to have this small presentation. Maddalena was already introducing an important issue related to the need of increasing sustainability in agriculture, in particular in viticulture, and the importance of soil. But I want to consider now another aspect that is related to the fact that our plants suffer from diseases. So we need to find strategy that can help to control diseases. And this is, in my opinion, another important issue for improving sustainability in agriculture, in particular also in viticulture, where we have many diseases that are affected. We know that in the next period we are facing really important emergency like the climate change, the loss of soil and soil degradation, change of different conditions, and new pests that can in some way affect our agriculture. So we need to be prepared for this type of emergencies that are actually taking all over the world, affecting all over the world also the agricultural system. Well, I introduce here the concept of the green revolution. Sorry, some words are in Italian. The chemistry has been really important in improving yield and safety of food. But since the book of Silent Spring from Rutger Carlson came out, clearly the awareness of the fact that the exceeding in chemistry, you are affecting soil, you are affecting environment, you are affecting also the safety of the food. So the need in some way, okay, it has been considered as a helpful thing for improving the yield and the quality, but we need to reduce the use of pesticide in agriculture. And so clearly, maybe there are different solutions. And I think that there is not only one solution, but many solutions that can be have to be taken in consideration in order to increase the efficiency of the production and to guarantee also for the next future the access to the food. I want to underline in this slide that in the last century we had a tremendous increase of the population, now reaching over 7 billion of people. So we need to feed all these people, and the estimation in the next century, in this century, is to further increase and to reach maybe 10 billion of people. So how we were able to feed all these people, how there are the effect of increasing quality of life, medicine, but also agriculture had a very strong impact in improving the food as quality, access, and safety. So feeding all these people. And chemistry, we have said that has contributed in increasing the field, the yield, mechanization, but also genetics. This is all concept of the green revolution that was described by Nobel Prize Norman Borlaug. I want to just focus on the genetics. Genetics, that it's another important tools with some conflict in terms of acceptance, but I want to say that it's remained an important tool to be considered. And we see in the history that humans have always improved plants with domestication, selection, and so on. Then in the last century, the impact in increasing yield and quality was really, really much higher. Let's say at least 10 or 20 more times more. But so in some way also contributing to increase, to solve the problem, to face the problem of the need of feeding more people. And this, in the last century, was mostly related to the development of the breeding techniques. So traditional breeding techniques had contributed a lot on this. Now there is a big discussion on the potential that all the new breeding techniques, the new biotechnology that are in some way always considered as from many aspects not so positive for the development. But now there are also new biotechnologies that can be considered more sure and can have a contribution. We need to probably understand better what are this technology and what can be the contribution of this technology for solving some of these problems. And I will say that again, the most important problem is to control diseases in order to reduce the food loss and reduce the use of pesticides. So no matter, in agriculture we have diseases and we need to protect plant from diseases, and we need to use pesticides. Also, and in my opinion, there is not a difference between a synthetic pesticide or a natural pesticide. Copper is considered a natural pesticide, but it's a toxic, heavy metals, that also in organic agriculture is used in high amount with an impact on the soil, for example, because it's accumulated in the soil, on the microorganism on the soil because it's altering the microorganism population. So it's really an issue for everybody to try to reduce the pesticide and to find solutions that can help to reduce the pesticide. Fungicides are the most common plant protection products. And for grape, it's really important that because more than 60% of the pesticide use in the world are used in the vineyard. So I would say that it's really an important issue for the future of sustainable viticulture. In order to reduce the pre-harvest losses and have a better quality and safety of the product. So what are the technologies? So this is technologies that we have in grape. So the clonal selection is a very traditional system for identifying new grape variety or selecting old grape variety and improving them through the phytosanitary control and characterization and so on. And then, and this is what is still the basic approach for viticulture, in particular in Europe, where the clones is really fundamental. So the importance of the clone for Italian and European legislation is very high. So we need to maintain the clone. So no, it's not very easy to modify. So in fact, breeding for introducing genetic resistance with interspecific crosses, that is something that is done and is offering a possibility for improving, for obtaining new variety resistance. It's quite easy to be applied in many different crops, but in grape, it has a limitation. This is an example of a work we are doing in strawberry, for example. You can use, if you apply this in grape, you have Vitis vinifera that is crossed with a wild Vitis grapes, like a Vitis riparia, for example, as a source of disease resistance. You need to do different backcross, but on the other hand, you have a new clone that is practically different from the original one. So it may be similar, but not the same as the original one. This practice is useful, and it is applied. New resistant clones are available on the market, but with this limitation, with the fact that if you want the original clone now modified because resistant to a disease, this technique is not offering you this opportunity. Another traditional opportunity is what we call the chemical mutagenesis. So the fact that you can apply mutagenic compounds that are approved, that are accepted, are standard, have been used since many years for inducing variability. Now these compounds can be applied in vitro. You can generate a new plant that are modified and then go to the field or in greenhouse and see if there is variability. This is the work that we are carrying out with some company, and we see that in the same clone, after this treatment, we can see single plants that are still having a very high susceptibility and others that are resistant. And this is the variations that we observe. So a small percentage is expressing a resistance. Clearly, this has to be tested. Maybe a new clone can be selected here with increased resistance. In this case, the new clone will maintain all the characteristics of the original one, but with this modification that has been induced with a natural treatment. This is not a GMO. This is natural mutation. I mentioned you about the new technology. Gene editing is one of the new technologies that can, in some way, replace the natural mutation with specific mutation. So what in a natural mutation occurs in, let's say, occasionally with uncontrolled system, with the gene editing, you can do the same mutation, but controlling it. And this, it's a possibility, is sure offering a possibility, and there is a big debate on the fact that this technology can be classified as a natural mutant, not as a new GMO. So consider this technology as a GMO. And this is a big discussion. Let's hope that people understand that this technology is very new and having very precise application specific in grape. And then another technology is the RNA technology that is based on the use of this small RNA, so nucleotides-based RNA, double-strand RNA, that has a specific capacity to target the gene. So are not new chemical molecules, are natural molecules that can be applied to control diseases. And so the work, for example, that we are doing always on grape, it's on botrytis, but also preliminary work are now for controlling Peronospora. We see that if we design a specific sequence of double-strand RNA that targets specific gene of the fungi, and we express stable expression of this thing, or we produce and we spray, and this is the difference between X and 6, this small molecule, we can protect the plant. And this is, I think that it is really a potential development for the next future with a new impact. Again, the discussion is, what do you prefer? Personally, I prefer this strategy, having an in-implanted stable expression of this small RNA with a very high specific target to the DNA expression, RNA expression of specific gene of the fungi. So have a stable resistance of the plant. Or produce and spray as standard spraying the plant with this small molecule. In the next future, I will say that you will have on the market many of these new products. Many new products are already pipeline, and so this will be something to discuss in the next future. This is GMO. This is not GMO. It's just a molecule that you spray in a plant. So I leave, and this is just to say work we are working with together with University of California, Riverside. They were showing for the first time that this small molecule silencing a specific gene was able to protect tomato, grape, and strawberry from the botrytis infection. So the potential of this new molecule can be very high because they are interacting with this. And in grape, it's the same. So we demonstrate that by spraying grape leaves, plants and leaves, we have a very strong reduction, and this was Cabernet, in botrytis infection. Well, all this work some of this work is already published, other is under preparation. The same also on the berries. Actually, this is technologies that can open also another issue, the fact that we can develop new products for that can help to increase the shelf life of the consumer. I finished. This is the last slide. Okay. So this is the thing. So my message was, we need to consider the genetic resistance as a potential application. We need to consider the new technology because they can help. We need to know better. In fact, my last message, the need to improve communication on the potential of these technologies for improving grape sustainability, and discuss together what can be the proper strategy for applying this and possibly get results that can help for growers and give more safety to the consumer. I finished. Sorry if I took a little bit more time.
Hi, I am Alejandra Núñez Verde. Let me share my presentation. We will speak about ship and wines. So we are here to introduce you an old and well-known material reconverted in a sustainable and natural packaging solution, just ship wool. Why ships? Because they have been producing wool since the ancient times. It's an amazing and natural, sustainable, and renewable fiber. For its insulation properties, it has been protecting ships and humans effectively for thousands of years. It is hygroscopic and it can absorb, maintain, and release humidity, improving climate control. Ship wool works as insulation even wet. It's also fire resistant and anti-static. It's resistant to compression and resilient and elastic. It's soft to touch and it does not cause irritation. But there are different kinds of ships. Coarse and rustic wool has all these advantages, but it has no textile value. This kind of wool is also a problem for a small producer because they need to pay for shear the ship and then bury, burn, or give the wool as part of shear payment. Considering these rural solid waste as a great raw material, we have created Abriga, a project that focuses on sustainability and circular economy. And we offer 3 families of products: Abriga, thermal and acoustical blankets, thermal and acoustical blankets for roof and walls, Acústica panels and furniture for acoustical comfort, and Envuelve, packaging solution, protective and insulated. Today we will present Envuelve, a sustainable packaging solution for protection and insulation. The packaging industry needs new solutions with environmental friendly material, which are natural, reusable, recyclable, and compostable. Envuelve combines all the techniques research we have for Abriga thermal insulation blanket with cutting-edge design. We can have different texture, natural ship colors, and natural dyed colors. Also, ship wool felt can be printed in serigraphy and could be cut in different shapes. So, Envuelve Bale is designed to protect fragile items such as wine bottles, replacing expanding polyethylene foam and bubble wrap, which accumulate a ton of waste every day. We can offer different thickness and densities according to each particular need. Envuelve bottle, a handmade bag for transportation that maintains temperature to a chilled bottle in different colors and could be printed with a logo or patterns in serigraphy. We offer not only the material and designed product, but also the possibility to co-create and produce with your brand new packaging solution and other creative uses of our technology. And this is our impact. Socially, we promote cooperatives with small family farmers and institutions by creating networks that collect and classify rustic wool for our project and for all other uses it may have, thus collaborating with rural roots and social inclusion. Economically, we create a new value chain based on fair trade with a small producer and developing local economies. And environmentally, we contribute to reduce solid waste and greenhouse emission by working with low embodied energy manufacture process with reduced carbon footprint. Wool in sheep also capture CO2 throughout their whole life. We collaborate with biodiversity, with regenerative agriculture and cattle raising. I am Alejandra Núñez Verte, architect, CEO, and founder of Abriga, with technical engineer Ernesto Benavides working with territory development. Our team is built by acoustic, industrial, and textile design experts, as well as social and circular economy experts working with us to create the Abriga project. In graphic design and communication, we work in alliance with Moorlander Design Inc., based in Toronto, Canada. This is our web page. Please contact us for more information and let's work together for a better world. Thank you very much.